Okay, hello. So as many of you may know already, climate change is a problem that is warming the temperatures of our oceans, which greatly impacts coral reefs, which are very temperature sensitive. And so the scientists of this paper were looking for ways to approach climate adaptation of coral reefs by looking at the genetic drivers of heat tolerance in coral. So their main question was that if they could use any given genotype from the coral to predict whether or not or how more or less they are tolerant to temperature increase. So this figure shows what the scientists did in the study. So they took coral from a coral reef of different specimens of the same species, Acropora millipora, which is shown in the image to the right. Um, and they took 7 samples, which they then cut into small pieces they call nubbins in the study, and they split all of those up into 4 separate tanks, two of which were control tanks, which they put, um, which were at a constant temperature, and the other 2 tanks were called stress tanks, in which they would slowly increase the temperature and study the changes of the coral within that tank. And what they were specifically looking for was the visual assessment of the health, because when corals are dying, they lose their symbiotic relationship with algae. You can see when they are bleaching and losing their color, also their photosynthetic performance, their symbiote density, and their chlorophyll content. And so they also used something called DART sequencing to assess the genotypes of the coral. DART sequencing is cool because after doing PCR to amplify the DNA of the coral, they can then use DART sequences to focus on regions of the genome that are less methylated, and the regions of the genome that are less methylated are the parts of the genome that have more variation. And so that is how they were able to compare and contrast certain SNPs in the coral, and correlate them with the heat tolerance that they were seeing in the coral. So this is the last figure of the study. And this figure is assessing the correlation between the genetic differences at certain points in the genome that they referred to as the heat-associated loci and the heat-tolerant phenotypes. So in the bottom square is their control, which is where they put in a bunch of random SNPs in the genome of the coral, and they saw that it does not show any correlation. But conversely, on the top half of the graph, they were using their heat-associated loci that they found with DART and correlating it with the heat-tolerant phenotypes, and they in fact did see a correlation. So here you can see that the lower the p-value and the lower the q-value, the higher the correlation, and the dotted line is the threshold of the significance. So yes, here at the bottom left corner is where the correlation is the strongest. Those specific genotypes can predict how tolerant a coral is to heat. And this is really cool and important because, especially for a lot of climate scientists who are actively working to rehabilitate and protect coral reefs, because it shows that genetic markers can connect corals' heat tolerance, which can allow them to breed specific coral in rehabilitation work, um, to further the likelihood that that coral will survive